Okay, imagine this. A fundamental idea in biology, something we've held on to for over a century, is now being seriously challenged.
Mm-hmm.
And that's exactly what seems to be unfolding right now in evolutionary science. It's got these, like, ripple effects across medicine, maybe even how we think life began.
It's huge.
So today, we're diving into some fascinating new research. It suggests that mutations, the supposed accidents that drive evolution, maybe they're not quite so random after all.
Indeed. For decades, the standard view, the prevailing view, has really been that these genetic mutations are purely random, just chance events.
Right.
And then natural selection comes along later, acts as the architect, sifting through all these chance occurrences.
Self the winners.
Exactly. But what if the chance of a specific mutation happening isn't actually completely independent of whether it's useful to the organism?
That's the core question here.
It is. And our mission in this deep dive is to explore these groundbreaking studies, look at the really clever methods they used, and unpack this new theory. A theory proposing there's an internal force, something intrinsic, working alongside natural selection.
And we're drawing on some really solid sources for this.
Yeah.
There's a recent study just published in PNAS, the Proceedings of the National Academy of Sciences, scientists from Israel and Ghana.
Yes, and also a detailed paper by Melamed et al., laying out the methodology and earlier findings.
Right. And these aren't just, dusty academic papers. They really offer a critical look at a concept that could potentially shift the whole paradigm. It could redefine how we think about life on Earth. So let's unpack these insights for you.
Let's do it.
So to really get why these new findings are such a big deal, we have to start with that bedrock assumption. The one that's like anchored evolutionary biology for more than 100 years.
Mm-hmm. The standard dogma.
Yeah, exactly. This core belief is that evolution is driven mainly by random mutations. These genetic changes just happen purely by chance. No connection to whether they're actually helpful or not.
Only later.
Only after these sort of genetic accidents pop up does natural selection step in. It acts like the filter, right? Picking out the ones that happen to be advantageous.
And that's always been the sort of, the tricky part with this traditional view.
How so?
Well, the theory itself was robust, made a lot of sense, but it's been technically impossible, really, to directly check if mutations actually originate randomly.
You couldn't just watch them happen?
Not really. Mutations are incredibly rare events, at the individual level. That rarity, plus some pretty significant tech limitations, just stop scientists from observing single mutations as they arose naturally. In real time, we couldn't track those tiny changes right at the source.
Okay, so it was a core assumption, but one they couldn't actually test directly.
Not until recently, no.
Yeah.
It was an assumption we had to make.
So this wasn't some minor detail we just glanced over. It was fundamental and untestable. Until now, apparently.
Exactly. Which brings us to a pretty significant breakthrough. Professor Adi Livnat, Dr. Daniel Melamed, and their team, mainly at the University of Haifa, they developed this new ultra-accurate detection method.
Okay.
They call it mutation enrichment, followed by upscaled maximum depth sequencing. Bit of a mouthful, so we'll call it MEMS.
MEMDIS, got it. And this is the game changer.
It really represents a major leap forward in our ability to actually probe the origins of genetic change.
So what makes MEMS so different? Why is it transformative?
Well, its big advantage is overcoming the depth limitations of older techniques. Things like duplex sequencing were good, but MEMS goes further. It lets them precisely measure individual mutations down to a single base resolution.
So like identifying a change in just one letter of the DNA code.
Precisely, and within incredibly tiny targeted sections of DNA, sometimes just like 6 base pairs wide. Imagine, finding a single typo, but you're looking intensely at just one specific 6-letter word in a whole library. It can even count these brand new mutations, de novo mutations, in human sperm samples.
Even if they're super rare.
Even when they're incredibly rare, like finding just 1 or 2 altered DNA molecules among millions, literally millions, of normal ones. That's a level of precision we just didn't have before.
Okay, that is impressive. How does it actually work, in simple terms?
Well, the key is that it combines high-throughput sequencing, sort of the standard workhorse now, but with this crucial mutation enrichment step.
Enrichment.
Yeah. This step is clever because it lets them effectively remove most of the normal DNA, the wild-type sequences that haven't mutated.
So you get rid of the background noise.
Exactly. By doing that, they can dramatically boost both the accuracy and the yield of their detection. It lets them focus right in on those extremely rare mutation events that were basically invisible before.
Okay, makes sense. Now, you mentioned this team, Milam et al. had earlier findings. This wasn't the first time they used MEMDs.
That's right. This isn't their first rodeo with this tech. They previously used MEMDs to look at a really famous mutation, the HBS mutation.
Ah, sickle cell.
Exactly. Many people know it. One copy protects against malaria, which is obviously a huge advantage in some parts of the world.
But 2 copies cause sickle cell anemia.
Right. A classic evolutionary trade-off.
So what did they find when they applied MEMDs to HBS?
Well, their initial findings were pretty striking, really surprising, actually. The HBS mutation did not seem to arise randomly.
Not random. How so?
It turned out it emerged significantly more often in the specific gene involved, the HBB gene.
Which makes the hemoglobin protein.
Correct. More often there than in a very similar gene right next door, the HBD gene. HBD is a paralogous gene, meaning it's related through evolutionary history, very similar sequence.
Okay, why does that distinction matter?
Right.
HBB versus HBD.
It matters a lot because it showed the mutation wasn't just popping up randomly anywhere in that region. It was specifically, or at least much more frequently, occurring in the gene where it actually provided that huge adaptive advantage protection against malaria in sub-Saharan African populations.
Or malaria is endemic.
Precisely. So this was a really profound early clue. It suggested something more than just pure blind chance might be influencing where mutations occur.
Okay, so if it wasn't pure chance for HBS, that must have raised some big questions for the team. Where did they go next? Did they immediately think, let's find another example?
Absolutely. Building on that HBS discovery, the logical next step was clear. Find another well-known adaptive mutation and see if the same non-random pattern holds up. See if it's a general principle or just a fluke with HBS.
Makes sense.
And that led them directly to the human APL1 gene. That's the main focus of the new PNAS paper, the one we're really digging into today.
APL1. Okay, what's the story with that mutation?
Well, its story is equally compelling. And again, it's another one of these evolutionary double-edged swords.
How so?
The APL1 mutation, certain variants of it, provide protection against a really nasty disease called trypanosomiasis. You might know it as African sleeping sickness.
Ah, yeah. Transmitted by the tsetse fly, right?
That's the one. Historically, it caused tens of thousands of deaths, particularly in Central Africa. So protection is a huge benefit.
But there's a downside, like HBS.
Yes. Similar to HBS, having 2 copies of this protective APL1 mutation significantly increases a person's risk of developing chronic kidney disease later in life.
Right. So another complex trade-off. Adaptation comes at a potential cost.
Exactly. It really highlights these intricate balancing acts in evolution.
So for the researchers looking at APL1, the central question must have been pretty similar to the HBS one then.
Precisely. If this specific APL1 mutation, the one giving protection, arises purely by random chance, then logically you'd expect it to pop up at roughly the same rate in all human populations around the world. But if its origin isn't random, if it's influenced by usefulness, you might expect it to arise more frequently in populations where it actually offers that protective advantage. Where sleeping sickness has been a major selective pressure.
Like in sub-Saharan Africa.
Exactly.
And what did the results show? Did the pattern hold?
The results, again, strongly supported the non-random pattern. It was quite clear.
Really?
Yes. The specific mutation they studied, it's called G1-1024AG. It arose significantly more frequently in the sperm samples from sub-Saharan Africans compared to samples from Europeans.
How much more frequently?
The rate was about 2.7 times higher in the African samples.
Wow, nearly 3 times higher.
Yeah. And critically, that's the population that has faced generations and generations of endemic trypanosomiasis, unlike Europeans who haven't had that specific selective pressure.
And did it arise in the right spot, genetically speaking?
Yes. Just like with HBS, it also arose preferentially in the precise genomic location within the APL1 gene, where it actually confers that protection against the disease.
So wait, these mutations, they're not just showing up more often in the populations that need them, but also, like, in the right place within the gene to be useful.
That's what the data strongly suggests. It's
Wow.
not just random noise across the genome. They seem to be appearing, or at least appearing far more often, right where they're most needed, evolutionarily speaking.
It almost sounds like the genome has a subtle preference or something, or maybe focus is a better word, focusing mutations where they might matter most.
That's perhaps one way to think about it, yeah. It's certainly a huge shift from just thinking about random scattering across the entire genome.
And the fact that they saw this pattern again with APL1, just like they did with HBS, that makes it much harder to dismiss as a fluke, right?
Absolutely. This consistency is key. Seeing the same non-random pattern, higher frequency in the relevant population, specific location across different genes, responding to different selective pressures like malaria and sleeping sickness, that's what really demands we reevaluate our standard evolutionary framework.
It suggests this isn't some weird anomaly. It might be a reproducible phenomenon, a general principle even.
That's the argument being made.
Okay, so this repeatable pattern, beneficial mutations, seeming to appear more often where and when they're needed, this obviously needed a theoretical explanation. Where did Professor Livnat go with this?
Right. This led Professor Livnat to propose a really fascinating new theoretical framework. He calls it natural simplification.
Natural simplification, okay.
And this theory moves significantly away from both the traditional idea of purely random mutation and also the old, long discredited idea of Lamarckism.
Right, Lamarckism being the idea that, like, a giraffe stretches its neck and then passes that stretched neck trait directly to its offspring through altered genes.
Exactly. The inheritance of acquired characteristics. Natural simplification isn't that. It's something different.
So what is it? What does natural simplification propose?
It proposes that evolution isn't solely driven by that external force we all know, natural selection, survival of the fittest. It suggests there's also a previously unrecognized internal force operating within the organism's genome itself, working in tandem with natural selection.
An internal force.
Yeah.
That sounds, well, really interesting. Can you elaborate? How does this internal force work? What does it do?
The idea behind this internal force is that as organisms repeatedly use certain genetic interactions or pathways over many, many generations, think of a common biological process crucial for survival that gets activated again and again.
Okay, like a response to a recurring environmental stress.
Could be, yeah.
Yeah.
The theory suggests the genome itself sort of learns from this repeated usage. It adapts its own structure based on this accumulated information.
The genome learns how?
Well, learns is maybe anthropomorphic, but the idea is this accumulated knowledge, the history of what's been useful, makes it more probable for mutations to occur that effectively streamline or hardwire those already beneficial, frequently used interactions.
So it makes the useful stuff easier to activate or more efficient?
Exactly. It simplifies the genetic regulation involved, making these successful pathways less dependent on complex, multi-step controls.
You used an analogy before, like a keyboard shortcut.
Yeah, it's a bit like that. Imagine a complex keyboard command you use constantly. Natural simplification would be like that command eventually becoming a single, dedicated key on the keyboard itself. It's been hardwired for faster, simpler access because it was used so often.
Okay, that helps. To make this even clearer for everyone listening, maybe we can walk through some concrete examples of how this natural simplification might actually play out at the molecular level.
Good idea. Let's start with something like gene fusions.
Gene fusions, where two separate genes basically merge into one.
Exactly. Traditionally, these have been viewed mostly as accidental, rare genomic errors, big mistakes usually.
Right, just random breaks and joins.
But the new view, looking through this lens of natural simplification, suggests something else might be going on. It suggests that genes which have evolved to be used together frequently over generations.
Genes involved in the same pathway, maybe?
Precisely. Those genes might actually be more likely to fuse together.
More likely. Why? How?
It's not random chance governing the fusion. Remember, the genome isn't just a flat string of letters. It folds up in complex 3D shapes inside the cell nucleus.
Okay.
This folding naturally brings genes that interact or are regulated together physically closer in space.
Proximity increases the chance of a break and rejoin event happening between them.
That's the idea. Their physical proximity, driven by their functional relationship, makes them more susceptible to the molecular mechanisms that cause fusion. So the fusion isn't just an accident, it's a consequence of their long-term functional relationship.
And the result is simplification.
Yes. It effectively simplifies the system. It hardwires a complex interaction, coordinating the expression of two genes into a single, ready-made genetic unit. Makes the whole process more efficient, potentially faster.
Okay, that makes sense for big structural changes like fusions.
Yeah.
But you said this principle also applies to the kind of small changes we saw in APL1 and HBS, the point mutations.
Exactly. The theory extends to those as well. It's not just about large rearrangement.
How does simplification apply to a single letter change in the DNA?
Well, one proposed mechanism involves RNA editing. There's a common process called A to I editing, where an adenosine A in an RNA molecule is chemically changed to inosine I. Functionally, I behaves like guanine G.
Okay.
The theory suggests that if a specific A in a gene is consistently being edited to I at the RNA level, meaning that A to G change is functionally useful and happening repeatedly, this active editing process itself might actually increase the probability of a permanent A to G mutation occurring at that exact spot in the underlying DNA sequence.
Whoa. So the temporary fix, the RNA edit, influences the chance of a permanent fix in the DNA blueprint.
That's the hypothesis. It's like the constant rehearsal of that beneficial change at the RNA level makes it more likely to become permanently encoded. There's also evidence linking transcription itself, the act of reading a gene to make RNA, to increase mutation rates in highly expressed genes, particularly in the germline.
So genes that are used a lot are more likely to mutate.
Potentially, yes, and in ways that might be relevant to adaptation. The genome's functional activity isn't just passive. It seems to actively influence the patterns of its own future changes.
Okay, this is really shifting my perspective.
Okay.
The core idea, then, is that what the genome does, its functional activity, directly influences the probability and location of future mutations.
That's the essence of it, yes.
It's not just a blank slate, passively waiting for a random lightning strike, a random mutation. It's an active, dynamic system, informed by its own history, its own accumulated information over generations.
Precisely.
So what are the biggest implications here? If this holds true, how does it change our fundamental understanding of life and evolution?
Well, the implications are potentially profound, really far-reaching. First, it reframes mutations. They're not just isolated, purely accidental blips anymore.
Right.
They become potentially meaningful events within a much larger, longer-term evolutionary process. It suggests the causes of mutations, the internal processes influenced by past selection, and their consequences, the potential adaptations, are meaningfully related.
But crucially, you said this isn't Lamarckism.
Absolutely crucial distinction. It's not Lamarckian because the environment isn't directly instructing the genome on how to mutate for immediate benefit. Instead, the internal information that biases mutation probability, the genome structure, its epigenetic marks, its editing patterns, that information has itself accumulated gradually under past natural selection. It reflects the organism's current structures and functions, which are products of past evolution.
Okay, so the bias isn't coming from the outside environment now, but from the inside, based on what worked before.
Exactly. It means the distribution of mutation rates across the genome at any given moment isn't random. It's specifically relevant to the adaptations that are likely evolving or being maintained at that time.
That's a huge shift. It feels like the genome is less of a passive reactor and more of an active participant, maybe even a collaborator in its own evolutionary story.
Yeah, that's a good way to put it.
So for you listening, this really challenges a fundamental principle of how we thought life adapts. It's suggesting that new genetic variations, the raw material for evolution, might arise not just from blind chance, but also as a consequence of mutations actively simplifying existing successful genetic regulation. This internal force, natural simplification, working alongside external natural selection, it paints a much more nuanced picture, potentially a more directed view of evolution.
Directed is a tricky word, but certainly less purely random. And this framework, this idea of simplification based on usage, it actually resonates outside biology too.
Oh, how so?
Well, think about that gene fusion mechanism genes used together become more likely to fuse. It strongly echoes a basic principle from cognitive science, from how our brains learn. It's called chunking.
Chunking, like remembering phone numbers in groups.
Exactly. When you learn complex information, pieces that repeatedly occur together, like the digits in an area code, eventually get chunked together in your memory into a single, more easily managed unit. It's a fundamental simplification strategy for handling complexity.
Huh. So the genome might be using a similar simplification strategy to the brain.
The parallel is certainly suggestive. It hints at maybe a deep underlying principle of how complex systems, whether biological or cognitive, handle information and become more efficient over time. This could have implications way beyond evolution, maybe for medicine, for computer science, artificial intelligence, even for thinking about how life originated in the first place.
It also makes me think about this concept you mentioned earlier, genomic friction points.
Where
These long-term selection pressures, like for malaria or sleeping sickness resistance, lead to specific mutational pressures. These double-edged sword mutations like HBS and APO1, providing protection but also carrying risks. They seem like perfect examples of these friction points, right? Highlighting this complex dance between adaptation, cost, or maybe this underlying simplification process.
That could very well be prime examples, yes.
Yeah.
Situations where the system is under intense pressure, leading to these non-random mutational outcomes that offer a benefit, albeit an imperfect one.
Wow. Okay, this deep dive has really unpacked a potentially revolutionary idea. That evolution isn't just a story of random chance filtered by survival, but maybe also a story involving an internal, informed process, a process of simplification based on history.
Mm.
So the next time you think about adaptation, maybe about how quickly bacteria evolve resistance or how species colonize new environments, you might just wonder, did nature have a little more foresight or at least internal logic than we ever gave it credit for?
It definitely raises a fascinating question, doesn't it? If mutations are, even in a subtle sense, directed or at least biased by prior accumulated information,
Hmm.
what does that imply? Does it say anything about the potential predictability of evolution, or maybe even our future ability to engineer specific adaptations more effectively?
Yeah.
It really opens up entirely new avenues for research. It asks us to fundamentally reconsider the very nature of biological innovation. Where does novelty truly come from?
A lot to think about. Thank you for joining us on this really fascinating journey today. We hope this deep dive has given you a fresh and maybe slightly mind-bending perspective on the intricate dance of evolution. Hopefully it left you with plenty to ponder.